So with lab 2, we'll begin to actually dissect our brains. For this lab, we'll be making a cut in the sagittal plane, but overall we'll be making cuts in 3 different planes of dissection. So you'll need to know these planes. The first plane is the horizontal plane, so this cut is made horizontal to our bodies. The second cut is the sagittal plane, and this is the cut that we'll be making this week. A sagittal cut is performed by cutting directly down the midline. So you're essentially splitting the brain into 2 equal parts when making this cut. Finally, the cuts that we'll be making during laboratory 3 are known as the coronal plane. These cuts would look like this on our sheep brains. So you have the coronal plane that would be made in this axis. Here's how you'd make a horizontal cut from the side of the brain. Our sagittal cut this week is made right down the midline like this. So let's take a look at how we're going to make this cut and some of the structures that will become visible to us. For lab 2, we're going to be making a sagittal cut on the brain. And this will allow us to separate the 2 hemispheres. So this cut, the sagittal cut, a mid-sagittal cut in particular, is going to be taking place right along the midline of the brain. So first of all, both hemispheres of the brain are connected together, and there is some meninges, particularly pia mater, the meninges that are flush with the surface of the brain that are holding the 2 sides together. So first what we're going to want to do is just very gently use the scalpel to cut away some of those meninges, just very gently applying pressure all the way to the caudal extent towards the cerebellum again. And that'll allow us just to look into the brain a little bit, get some of those meninges out of the way. Now, using either a scalpel or I'm going to use a knife, we're going to make one clean cut right along the midline. So you don't want to use a sawing motion. That's going to destroy some of the brain structures. You want to make just one nice clean cut right along the midline. So I'm going to turn the brain over and then just make one cut, just applying pressure right down the middle of the brain. Okay, so now that we've made our mid-sagittal cut, we can actually start seeing some structures that we couldn't see before from the external surface of the brain, structures that are embedded inside the brain. And a lot of these structures are going to be what are known as the between brain or diencephalon. It's between brain, right, because it's located between the forebrain, where the cortex is, and the brainstem. So this is all between brain. And right in the middle, right here, is this large sphere, right? This is the thalamus, right here. Just below or ventral to the thalamus, we have the hypothalamus. And then posterior to the thalamus is the pineal body. And Descartes actually thought that this was the seat of the mind right here because there's actually only one pineal body in the brain, unlike most of the structures that we'll see. And you can see that the pineal body, you can actually just remove this all the way from the brain there, this little sphere. Then some other parts of the brain that we couldn't see before, right? We saw the pons and medulla brainstem, but now we can actually visualize some of the midbrain brainstem. This includes the superior colliculus. Then just below the superior colliculus, we have inferior colliculus. You can see now the inside of the cerebellum. Finally, a really important white matter structure. You'll be able to see this really intense white matter. And this is the corpus callosum right here. And you can see that it makes this almost inverted C shape or inverted U or C-like shape. And this white matter is connecting both hemispheres together here. There's a little bit of space right next to the corpus callosum, and these are the ventricles. Particularly here's the lateral ventricle. Then you have the third ventricle surrounding the thalamus. So So these are some of the structures that we'll be able to see. In addition to those, there'll be some other ones that are outlined in your LabTube protocol. Once you're finished identifying your structures, you're going to take a paper towel, spray this down with some of the Carolina wetting solution. This is going to keep our brains nice and moist. You can then wrap up your brain within the paper towel. And this will all be stored within your Ziploc baggies. Here we have our meninges right in our bags, and then we're also going to place all of our brain matter into our Ziploc bags to keep it nice and fresh.